Yeah, okay. I'm going to talk about development of gene therapy for Gaucher disease, and I will only talk about experiments that we've done in our own lab using experimental animals. And I mean, there's maybe no need to show this slide because Tim Cox gave an excellent overview of the background for Gaucher disease. It's basically an enzyme deficiency, as you know, and there are many different mutations, over 300, although, you know, 2 or 3 of them are the most frequent, as the previous speakers have pointed out. Now, basically, there's an accumulation of the substrate, glucosylceramide, that leads to accumulation in the Gaucher cells. It's basically macrophages that are lipid laden with the substrate. And clinically, it needs to splenomegaly, hepatomegaly, cytopenia, and bone disease in many cases. Now, I will focus only on type 1 Gaucher disease, which is the type of the disease that I've been trying to develop gene therapy for, but the same might be able to apply also to type 3 disease. And as the previous speakers have pointed out, there are existing therapies that are working quite well. The enzyme replacement therapy developed by Roscoe Brady has been working very well, but it requires intravenous treatment of the enzyme. The disadvantage is it's not curative. And then there is a substrate reduction therapy, reducing the synthesis of the substrate. And the good thing is that it's an oral therapy, but it's just like the enzyme replacement is not curative. And then there is allogeneic bone marrow transplantation. Now, this is practically never done at the present time, but it was developed in the 1980s and perhaps into the early 1990s. And the reason why people don't do it, despite the fact that it's curative, is some severe complications that can occur. Graft versus host disease can be quite severe, and that's why this therapeutic approach is not used. However, gene therapy would be based on a similar system, except then you're using the cells from the patient, him or herself, and that means that you don't get any graft versus host disease. So it would be promising to be able to develop a gene therapy of hematopoietic stem cells and transplant, and this I will talk about. Now, this is just an overview over the model that we generated for mouse model for Gaucher disease. You see here that we remove these 3 exons in the gene, and that leads to an allele that cannot produce a functional enzyme. And the consequence is that we have accumulation of the substrate glucosylceramide in bone marrow, spleen, and liver. And here you see on the left a normal bone marrow, and here is the bone marrow from Gaucher mice, and you see all these big lipid-laden macrophages or Gaucher cells. So also over time, the spleen grows substantially. So at 16 months of age, these mice, they have spleen size that's about maybe 10-fold, what it normally should be. And the main aim of our study that we have actually already published this, but it was to test a clinical grade lentiviral vector that doesn't have any GFP marker gene. So, that we've used an EF1 alpha short promoter to drive the glucocerebrosidase gene, and the aim was to enter clinical studies, and they have already started. So, this work was done in collaboration with AvroBio. So we helped them for some years to test this vector in our mice and showed that it works. We also some toxicology studies were done. I'm not going to talk about that. Now, I just want to mention this company, AvroBio, and they basically did get permission to test clinical gene therapy in Gaucher patients, and they've done it, gotten it in Canada, and I think also in Australia. So I'll come back to this. Now, this is the vector that we used and that the company has also used with the EF1 alpha short promoter driving the glucocerebrosidase cDNA. And we tested two approaches. One we called early intervention. That was, we induced the disease. That's to say, to make the deletion so that the disease starts developing. And we waited only 11 to 16 weeks here. And then we did the gene therapy, transplant into irradiated animals, and then analyzed them 20 weeks after the gene therapy to figure out whether this had been effective. What we call late intervention, which is a more important and more stringent test. Then we induced the disease, waited 6 to 9 months until the disease was really quite developed in the animals, and then we did the gene therapy and transplant into recipients, and again analyzed them 20 weeks post-transplantation. The reason why you choose 20 weeks is that at that point it's assumed that all the cells in the blood system are derived from the stem cells that were used to transplant the animal. And so these are findings from the early intervention study when the animals were not that sick at the time of therapy. But we see that if we look here at the glucosylceramide, the substrate accumulation, when we have MOC or no therapeutic vector, we have high levels of the substrate, but with a lentiviral therapeutic vector, this level of substrate has reduced substantially and is frankly quite close to what it is in the wild type. This is in the bone marrow. And if we look at the spleen, you see practically the same thing here. That's very prominent reduction in the lentiviral vector treated animals. And then in the liver also you see the same thing. There is a very good reduction here in the lentiviral vector treated animals. Now this is the late intervention study where the animals were sicker when they were treated, and you can see here that they have a very high level of, in untreated mice, a very high level of glucosylceramide. And there's a substantial reduction in the lentiviral vector treated animal. However, this reduction is not as prominent as it was in the early intervention study. However, we can expect that if we would look at these mice like 4 or 5 months later, then this value would go down. In the spleen, we have a very good reduction in the substrate in the lentiviral vector treated animals. And the same is true in the liver, where we have a very good reduction compared to the untreated controls. Now, this just shows the blood parameters, and I'm just going to focus here on the hemoglobin. Now, our mice, they have a mild anemia, as shown here, that the somewhat lower hemoglobin value than in the normal mice. But in the lentiviral vector treated animals, the anemia has disappeared and the hemoglobin value is more or less normal. And the same thing is for the hematocrit, which here is normalized in the vector treated animals. Now, this shows the hepatosplenomegaly in the animals. This is the spleen. And so in the vector treated animals, the spleen size is significantly reduced and is almost to the level that you will see in wild type animals. And the liver, you see also a significant reduction in the size of the liver in the vector treated animals. And this shows the pathology, and in the animals that didn't receive a therapeutic vector, you can see all these Gaucher cells here in the bone marrow and also in the spleen. Whereas in the lentiviral vector treated animals, the bone marrow is clean and it's very similar as to in normal mice. And the same is true about the spleen, also similar to the normal. And you can see here the red and white pulp. This is a normal spleen structure. This is just a slightly higher magnification, makes it easier for you to see that here in the untreated animals, you see lots of Gaucher cells, whereas these have disappeared in the lentiviral vector treated animals, and that's similar to the wild type normal animals. Then we did some bone studies, and this was done very much at the initiative of AvroBio, and they looked at mean bone mass and bone density. And you can see here that the bone mass, there is a significant increase in the treated animals, shown here by number 6, group 6, compared to the untreated animals here. The same is true with the density, except that it doesn't reach statistical significance. But this is actually very encouraging, that the bone pathology seems to be improved by gene therapy. Yeah, this is a bit of a complicated slide that you have to have if you are publishing a gene therapy paper. It is to look at the integration sides of the vector. And please ignore the top part of the picture, but the lower part shows individual animals, and each color represents a integration site. So this one has mainly 2 integration sites, and this one has slightly more. What's encouraging is that the integrations are mostly in transcriptional, in the transcriptional part of the gene, and they are not at the transcriptional start site to disturb the regulation of the gene. And there is no difference between the late and early groups. And most animals, they show what you call an integration pattern showing only oligoclonal integration, not so many integrated clones. This is typical for mice, also shown by other vectors. And most importantly, at the bottom here, there are no integrations in or near high-risk proto-oncogenes. So that's the most positive finding. And so this seems to be a relatively safe integration pattern, if you like. So just to summarize, what I've said is that when we have used a clinical lentiviral vector to treat type 1 Gaucher mice, and there is a robust and significant reduction in glucocerebroside accumulation in bone marrow, spleen, and liver. And the levels are close to those in normal mice 4 months after treatment. And the histopathology showed the practical disappearance of Gaucher cells in the marrow and spleen. There was a reversal of hepatosplenomegaly. There's also that the blood parameters, hemoglobin and so on, were normalized in the treated animals, and the vector integration showed a typical lentiviral integration profile, and there were no integrations in high-risk proto-oncogenes. So just to mention the one thing that's important and maybe the highest risk in this kind of treatment, and that is you have to use chemotherapy to get good engraftment of the transplanted and treated vector-treated stem cells. And in other diseases, busulfan or melphalan in a lower dose, much lower dose than you use in allogeneic transplant, has been used safely. And so the conditioning gives space for the new cells that can home and engraft in the bone marrow niche. And it's busulfan that has been used in the clinical study. So the stem cells have been mobilized by growth factors from the patient, and they are harvested in the peripheral blood in an apheresis machine in the blood bank, and then purified and expanded and transduced, meaning the gene transfer takes place here. And then by giving low-dose busulfan conditioning, the engraftment becomes more efficient, and the treated cells now home and engraft in the bone marrow. Just want to mention that AVROBIO is a company that we helped develop the clinical trial, but unfortunately, the company has not involved me directly in the clinical trial, so I'm very poorly informed. I did contact them 2-3 months ago, and then apparently the first patient they had treated had been going for 6 months, and there had been no serious complications, and the numbers they claimed looked good. However, I didn't see them, so I can't tell you how well the gene therapy was working in that case. They had also started treatment of 2 other patients, although they were not as far gone. So it's very much up to AVROBIO to report to people how this is going, and I think they should do that, particularly the people like you with disease associations who are obviously very interested to know how this trial is going. And finally, I want to thank all my colleagues who have helped me with this, and primarily Maria Dahl and Emma Smith, who did most of the experiments with the clinical lentiviral vector. And I want to thank AvroBio, the company, so they financed these studies. Chris Mason is the chief medical officer, and particularly valuable have been people from Hannover Medical School who designed the vector and who do all the insertional vector integration analysis. Axel Schambach, a long-term collaborator over 10, 15 years. And Hans Ertz helped us with the lipid analysis, and AvroBio hired Charles River to do some of the toxicology studies. So that's it, and that's the end of it. So I'm happy to answer questions if there are any.
Yes, I had a question. How long do you think before they could do any medical studies on type 3 patients?
Well, I maybe can't tell you, but as Tim Cox mentioned, the AvroBio company, they are planning to start such a study in 2023, which is next year.
Okay.
I think this is a highly relevant question because I can't see why we shouldn't test type 3. I think it potentially can help a lot of type 3 patients. So I hope that they will get going with that and be able to answer that question.
I wonder if I could just say something slightly mysterious, Stefan, on that. In the first patient who was treated had a genotype that many people in Sweden would recognize.
Yeah.
Okay.
And it's worth following that up in some detail, in fact.
Yeah.
I always thought actually that a very easy role would be in type 3 disease for various reasons that we understand. And so, but as I say, the very first patient, sorry about the connection, the very first patient, I think it is in the public domain, but the genotype of the first patient might give some feeling for encouragement.
Okay.
In that direction. The type, yeah.
Were there any other questions? Okay, so then thank you very much, Stefan Karlsson, for your presentation. And